Thank you, George, for that fantastic introduction, and thank you for giving me the opportunity to talk to you all today about my work with NanoSIMS. So I thought it would be useful to talk to you today about NanoSIMS. It's not a technique that's particularly well known, so I thought I would spend a little bit of time talking about what it can do, what it can't do, and as George mentioned, really focusing on how we've been using it to look at hydrogen and deuterium in a range of different metals. So first of all, NanoSIMS is based on the principle of secondary ion mass spectrometry, or known as SIMS. So with this technique, we use a charged, focused primary ion beam to bombard our sample surface. And this results in removal of the top few atomic layers of our sample surface in a process that is known as sputtering. And in that sputtering process, we generate a range of different species that come off of our sample surface that range from secondary ions to neutrals, molecules, and resputtered primary ions. It's actually only the secondary ions which we can analyze and collect. So we collect those secondary ions and we send them into a mass spectrometer. And what that allows us to do is to analyze those elements according to their mass. And therefore, by scanning this primary ion beam over the sample surface, what we can do is we can build up a chemical map of the elemental distribution inside our sample. And it's worth noting as well that this really is a very surface sensitive technique. We're really only sampling the top few nanometers of our sample surface in this analysis. So here we have a very simple schematic of the NanoSIMS, how it's laid out. So on the NanoSIMS, we have two primary ion beams, a cesium plus ion beam and an oxygen minus ion beam. And that is what we use to focus down onto our sample surface. As I said, we scan that primary ion beam over the sample, generating those secondary ions which we want to look at. We extract those secondary ions through a series of optics, and then they enter a very large magnetic field, or a large magnet, I should say, into a magnetic field. And that magnetic field separates out the secondary ions according to their mass. So the magnetic field will affect lighter elements more strongly and heavier elements will be affected less strongly. And that's how we can separate out all these ions as they come off our sample. So on the NanoSIMS, we have 7 detectors. And so by positioning those detectors at the right position where those elements are coming out of the magnetic field, and by scanning the primary ion beam, we can therefore build up chemical maps of our sample. So this is an example here of a cell that we're imaging just here. So this is how the NanoSIMS looks at the University of Manchester. So at the top of the instrument here, we have we have the oxygen source here, and we have the cesium source just below it. The primary ion beam comes down through the microscope at this point and is sent towards the analysis chamber just here. So our sample is sitting in here. Secondary ions then travel back through this route here. Here we have the large magnet at the back, and in this chamber here, we have the detectors positioned where they need to be. Now, it's worth noting that the NanoSIMS operates under an ultra-high vacuum at about 10 to the minus 10 millibars. So that's much higher than you'd expect to get in an SEM or TEM. And because of that, we have quite a lot of pumping capacity at the start here. So we have the airlock here where we introduce our samples into the NanoSIMS. We then have a storage chamber just here, and the analysis chamber is just here. Now at the University of Manchester, our NanoSIMS is actually used for a very, very wide range of samples and across a wide range of disciplines. It's very diverse. I'm just going to be talking mainly about hydrogen deuterium detection here, but I thought it was worth pointing out the range of what we can actually do with a NanoSIMS instrument. So this just gives you an idea of some of the experiments that we've been working on over the past few years. So what I'm going to talk to you about today is mainly related to hydrogen embrittlement in metals and trying to be able to detect hydrogen deuterium within them. As mentioned in George's introduction, a lot of the work that I did previously was related to elemental uptake of elements into crops and plants, whether that's for nutrition purposes or we're looking at where toxic elements end up inside, for example, rice grain. We do a lot of work, of course, with advanced materials here in the department and also through the Henry Royce Institute. My colleagues in earth sciences use the NanoSIMS quite frequently to look at natural isotopic distributions in extraterrestrial materials. And so by this, I'm referring to things like space dust and meteorites. Again, with colleagues in earth sciences, we've also used the NanoSIMS to look at the metabolism of bacteria and how they interact with particular minerals. And we're also starting to now look at drug localization. So where does the drug end up within the cell? So here we have a cell nucleus, for example. Is the drug targeting the right part of the cell to actually perform its function? So I also thought it would be useful to run through a few of the capabilities of the NanoSIMS and try and understand why it can be used for such a wide variety of topics, but also why we're using it to look at hydrogen distribution in metals. So these are the key characteristics of the NanoSIMS, and I'm going to talk you through each of these now. So one of the key capabilities of NanoSIMS compared to other SIMS instruments is the fact that it has high lateral resolution. So we can focus that primary ion beam down to 50 nanometers. And this is much higher than you could get with other SIMS instruments. It's also higher than you can get with chemical mapping in the SEM, for example, but not as good as TEM, for example. So it fits somewhere in between. Now, routinely, we typically don't image samples at 50 nanometer resolution because there's always a trade-off between resolution and sensitivity. So the NanoSIMS can actually achieve parts per million sensitivity for some elements, but we can't have that alongside our 50 nanometer resolution. So typically for a lot of our experiments, we're working in a range of between 100 and 200 nanometer spatial resolution, but that's still very good. It allows us to see, for example, subcellular information when we're looking at biological samples or distributions of elements at the scale of the microstructure. The high mass resolution capability allows us to distinguish between species that have nominally the same mass. So for example, at mass 32, sulfur has a mass of 32, but also two oxygen atoms bonded together also have a mass of 32. But the clear high mass resolution of the NanoSIMS allows us to separate out those two species very clearly, so we have confidence that we know exactly what we're looking for in the NanoSIMS. As I mentioned before, we have 7 detectors, so we can detect 7 ionic species simultaneously. And this is really important if we're trying to correlate a particular element with another element that tells something about the structure of our sample, so we can be sure where that element is located. We can also detect almost all elements in the periodic table, and that is right through from hydrogen all the way through to uranium. So there are very few techniques that can actually detect hydrogen, and even fewer that can spatially localize it. And because this is a mass spectrometry technique, we can actually detect isotopes of particular elements. And we can do this by either looking at natural variations of isotopes, as I mentioned in meteorites, for example, but we can also deliberately introduce enrichments of naturally occurring isotopes that have very low abundances. So we can track the uptake and incorporation of elements over time. And so commonly used isotopes that we use in the NanoSIMS are listed here. And in this particular presentation, I'm going to focus a lot on how we've been using deuterium, but I'm also going to show you an example of oxygen 18 as well. Now, no technique is perfect. The NanoSIMS does have some clear disadvantages to it, and I think it's worth highlighting these just to sort of manage expectations and perhaps preempt a few questions at the end as well. So, although we can detect almost all elements in the periodic table, there are some notable examples which are very difficult for us. So specifically noble gases, they are almost impossible for us to look at, and other elements such as cadmium and zinc simply don't give very good secondary ion yields. So it's very difficult to get enough sensitivity in order to be able to create an elemental map from those. The NanoSIMS is a high resolution technique, so we're not aiming to map over large areas of the sample here. We really need to try and concentrate on smaller areas and image those at high resolution. We can image over large areas, but it's quite challenging and it can be quite time consuming as well. So it's not the right technique for that. If you want any chemical state information, you're probably better off going to look at X-ray photoelectron spectroscopy, for example. And you may have come across SIMS before and know that it can provide molecular information. That is a slightly different kind of SIMS instrument you need for that. That'd be a time of flight SIMS. The NanoSIMS tends to give just elemental or isotopic information. Quantification is difficult. So what I mean by that is it's very difficult for me to say at a particular point on the sample surface, what is the percentage of elements that you can get from that. We can look at relative amounts, but I can't say you have 3% of your particular element in that particular location. We do not have a cryo stage on the NanoSIMS, and this can be an issue for lots of different application areas. Biology is a notable one. It means you have to go through some quite complicated sample preparation to get your sample into the ultra-high vacuum on the NanoSIMS. But specifically for this talk, hydrogen is a very mobile element, and we had a lot of concerns when we started off looking at these samples is, would we be able to find any hydrogen nanomaterials because we cannot keep our sample frozen and immobilize that hydrogen. I've also written in brackets at the end that this is a destructive technique by its very nature. You saw from that schematic at the start, the ion beam is removing material as we analyze it. So after you've conducted your analysis, you will have a crater on your sample, and that piece of sample will be gone. You cannot go back and analyze it again. So if you want to do a correlative or complementary study, including NanoSIMS, you need to make sure that NanoSIMS comes towards the end of that particular analysis program. But I put it in brackets because actually this can also be advantageous for us. Because we're removing material layer by layer, what we can actually do is see how distributions change as you go down in depth. So you can make a depth profile through your sample, or if you're imaging, you can create a 3D reconstruction of your sample to see how elemental distribution changes with depth. So what I want to focus on for this particular seminar is how we've been using the NanoSIMS to look at hydrogen embrittlement. So I'm sure a lot of you are aware, but just to briefly go through it, hydrogen embrittlement is where we get a reduction of toughness and load-bearing capacity of a metal once we get hydrogen entering into it at significant concentrations. And that hydrogen can cause cracking and catastrophic brittle failure of that material at a value that is much less than the expected yield stress. So if we look at this graph over here, we can see a stress strain curve. When we look at this nickel alloy being tested without hydrogen, it fails here. But as soon as you've got hydrogen introduced, it fails much earlier. And you'll see in these images here, it also fails with a brittle fracture surface as opposed to the expected ductile fracture surface. So the aim of these projects that I'm going to talk about today was to identify where hydrogen is being trapped in the microstructure of steel, nickel, and zirconium alloys by investigating the hydrogen distribution at high lateral resolution using the NanoSIMS. Because although we know the hydrogen is there and we can see the effects of it, we don't know exactly where it is located in a lot of cases. So there's not been a whole lot of work done on localizing hydrogen in metals. And one of the reasons for that is that there are very few techniques capable of even detecting hydrogen in the first place. A lot of those are in fact bulk techniques that do not give any spatially resolved information. And so localizing it to very specific features is also very challenging. Furthermore, when we're thinking about NanoSIMS experiments in this area, there's always ambiguity regarding the origin of the hydrogen. So did the hydrogen that you're detecting in the NanoSIMS come from your experiment where you deliberately introduced hydrogen into your metal through a charging experiment, for example? Or could it have come from the vacuum system? Although we're looking at very, very low vacuum levels in the NanoSIMS, there is still residual hydrogen in the NanoSIMS. It's very difficult to pump that out. Could it have come from sample preparation as we're polishing and grinding our samples in water, or just simply from storage by having water vapor in the air, for example? So an example I'm going to show you now of the issues associated with hydrogen and some of the artifacts starts to highlight that difficulty. So this is an image here of a zirconium alloy. This is in cross-section. The zirconium alloy has a relatively thick oxide layer attached to it, as you can see just here. And then over on this side, we have the underlying metal. Now, zirconium alloys tend to form hydrides as they're taken out of service. And we can very clearly see from this hydrogen map here, very clear presence of hydrides along this region here. But we can also see over on the left-hand side, we've got this somewhat smudgy region of hydrogen that's a little bit more difficult to explain. So did this hydrogen come from the interaction of this metal with the conditions in the autoclave where this sample was oxidized, or is it an artifact? Now, when we look at the carbon map that we acquired simultaneously, we can start to understand things a little bit more clearly. So what I didn't say is that over on this side here, this is resin. We mounted this sample in a resin so that we could polish it and hold it in the sample holder. And an image such as this may take half an hour to 1 hour to acquire. So as we're imaging pixel by pixel from the top here, hydrocarbons from the resin have enough time to diffuse over the sample surface. And so these hydrocarbons, as they diffuse across, they're bringing in hydrogen with them. And as I said right at the start, this is a very surface sensitive technique. So we're imaging now those hydrocarbons on the surface rather than the underlying metal. So because of this issue with detecting hydrogen, what we actually do is we image deuterium, and we artificially enrich the amount of deuterium in our samples. And I will show you how we do that very shortly. Now, the reason we're interested in looking at deuterium and using deuterium in our experiments is that the natural deuterium abundance that we find in everything around us is only 0.015%. So very, very low levels. So if we're putting our sample into water containing 50% deuterium, for example, then any deuterium that we detect in our sample pretty much comes from our experiment only. So you can really see this when we start looking at the deuterium map now. So now we no longer have that smudgy region. We only have deuterium being detected in the hydrides. And so we know that these hydrides must have formed from hydrogen that entered the zirconium during service. So everything that I'm going to show you during this presentation related to deuterium or hydrogen distribution in metals will be looking at deuterium maps. So how do we get that deuterium into our alloys in order to be able to investigate its distribution? Well, we use slightly different approaches depending on the metal that we're interested in. And we do that to try and get as close as possible to in-service conditions. So for the steel and nickel alloys, what we do is we use galvanostatic cathodic polarization. So here we have our steel and nickel sample in a deuterated solution, and we are charging that sample in ambient conditions in order to push deuterium into that sample. The zirconium is slightly more complicated. So zirconium is used as a nuclear fuel cladding. So the uranium fuel will be in the middle of these zirconium tubes, and the zirconium and the fuel will be placed into the nuclear reactor, and that water is going to be at high pressure and high temperature. So we're trying to simulate the nuclear reactor conditions as close as possible. So we place the zirconium tube, first of all, into an autoclave containing water with natural levels of deuterium. We allow the oxide layer to grow on the surface to a predefined thickness in most cases, or for a particular time. And then what we do is we take that zirconium tube out of the autoclave, place it into a different autoclave, and this time the water will contain elevated levels of deuterated water or water containing oxygen 18. So those are the approaches that we need to use for this experiment. So the first set of work that I'm going to show you was all conducted by a PhD student in my group, Dr. Yasser Abora. And Yasser's PhD project was to really try and set out how we could have confidence in our NanoSIMS results of hydrogen and deuterium. So there were a lot of artifacts that we knew might be at play here, trying to understand what was real and what was not real in terms of the distributions we were seeing. So the first issue that I want to highlight here is what is known as the matrix effect in SIMS. So here, what Yasser started working on first was a 303 stainless steel. And the reason we chose this alloy was because if you read the literature, it's very clear in the literature that manganese sulfides are supposed to be the location where hydrogen is trapped. So we thought, great, that will give us a very clear feature we can look at in the NanoSIMS. They're easy to find, and we should be able to see deuterium localized to these manganese sulfides, and we'd get a sort of benchmark of what we can and can't do from that. So here we have our hydrogen map, our deuterium map, our oxygen map, sulfur, which as expected shows a lot of sulfur in the manganese sulfides. And at the same time as getting the ion images, we can also generate a secondary electron image in the NanoSIMS, which as you can see shows the microstructure of our alloys very clearly. So when we look at our hydrogen and deuterium maps from this alloy, you can see up here, this region looks like it contains elevated levels of deuterium. But why should that particular grain have more deuterium than, say, this one here or this one here? Well, in fact, it doesn't. This is the matrix effect. This is what we see when we have different crystallographic orientations being imaged in the NanoSIMS. So some particular grain orientations will emit more secondary ions than others. So we obviously need to be able to account for that when we're looking at our images, because that obviously complicates our interpretation. And what we do is we simply take a ratio image. So we take a ratio of deuterium divided by hydrogen. And what you can see in this image is that it's basically flattened out the background in this case. We've now lost all of that grain orientation. And what's become really apparent is where deuterium is localized or enriched over hydrogen distributions. So essentially this image now shows us the locations where deuterium has been incorporated into the microstructure. So what I'm going to show you throughout this presentation will be ratio images rather than simply just the deuterium image in most cases. So for this particular steel example, you can see that the deuterium is localizing to dislocation slip bands. You can see we've got quite a deformed microstructure in this as received steel here. So we can see very clear enrichments of the deuterium. And interestingly, and we're still working on this, we don't really see any deuterium localized to the manganese sulfides. So this was really not what was expected according to the literature. So we need to think about that a little bit further. So as I said, Yasser's project was to really try and look at what we could understand from the NanoSIMS and what we could believe and what we couldn't believe. And I also mentioned at the start that we do not have a cryo stage on the NanoSIMS. So one of our major concerns was that before we could even analyze the sample in the NanoSIMS, all of our deuterium would have disappeared into the vacuum system. So Yasser set up an experiment using what we have added to the NanoSIMS, which is called a residual gas analyzer. This residual gas analyzer allows us to look at the composition of the vacuum. So although we've got a very, very good vacuum, there are still some residual gases remaining in there. And I'm not going to go through the whole of this graph, but I just want to highlight what is happening in this region here. So Yasser did a series of experiments where he put sample holders in and out of the chamber that has the residual gas analyzer attached to it and looked to see how the deuterium concentration in the vacuum increased and decreased. So at this point here, point B, Yasser introduced a sample that was charged with deuterium, a steel sample that was charged with deuterium into that chamber. And you can see the deuterium signal in the vacuum chamber rises very dramatically. Starts to get pumped away, and then as soon as that sample is taken out of the chamber, our deuterium signal drops off dramatically. So what that tells us is that deuterium is outgassing from our samples in the NanoSIMS while we're trying to analyze it as well. So the key thing was to try and get the sample charged into the NanoSIMS, pumped and analyzed as quickly as possible before we lose too much deuterium from it. So schematically, this is shown over here. If we have a charged sample, we can see that deuterium is escaping from our sample. We usually load multiple samples into the NanoSIMS at any one point. So in many cases, we would have a sample that we didn't charge to act as a control. And we found we could never measure the natural isotope ratio of deuterium, 0.015% from an uncharged sample, because we were getting deuterium depositing on all surfaces inside the NanoSIMS. So this has implications for measuring uncharged samples, but it also means that any part of our sample that does not contain deuterium from our charging, we would never be able to measure the natural isotope ratio. So it has an effect on our detection limits. In hindsight, we actually think that this may have been somewhat beneficial for us, because if we had frozen our sample and analyzed it in cryo conditions, all of the deuterium that we had charged into our sample would have still been present. We may have only seen a uniform deuterium image on the top surface, as all the deuterium would have still been there in the bulk. But actually, by allowing the sample to outgas a little bit, what it has done is it has improved our contrast. And it's actually left behind the deuterium that's in the trap sites, which is what we wanted to investigate anyway. So we may have actually been very lucky here by not having a cryo stage. So I now want to show some work on the nickel alloys that Yasser was looking at. So we were first looking at this alloy 625 plus, which was of interest for BP, who funded this project. And the first thing we did was to look at the as received 625 plus. If we look at the deuterium over hydrogen ratio image, we do not see any localized regions of deuterium. So we have no deformation and we see no localized deuterium. So then what we did is we improved, or we increased the complexity of our experiment somewhat. So now we were looking at a pre-strained 625 plus sample. So we took our as received sample and we strained it to a total deformation of 1.5% to introduce some strain into our material. So here in our backscattered electron image, which was taken before the NanoSIMS analysis, you can see that there are some regions where we have localized strain, specifically at these grain boundaries here. So when we look at this exact same region in the NanoSIMS and we look at our ratio map, you can see that we see small enrichments of deuterium at these localized strain regions just here. So where we've got a deformed microstructure with dislocation slip bands and really highly localized deformed regions at the grain boundaries, we can now start to see localized deuterium. So we're seeing a relationship between dislocations and deuterium. So then we increase the complexity again. So this time we were looking at in situ charging and cracking at the same time. So now this is a sample that's been fatigue pre-cracked with these conditions here, and all the conditions for charging are shown over here as well. Here we can see we have a crack running through this sample surface here, intergranular crack to a transgranular crack. We also get some surface cracking due to the charging conditions used here. And in this particular alloy, we can see we have sigma phase here on the grain boundary, which was of interest for us as well. So now we can see we have deuterium localizing to twin boundaries. We can see we have deuterium localizing to dislocation slip bands. And we can also see deuterium localized to sigma phase as well. So all of these regions show the highest deuterium enrichment. So what we've got now is deuterium transported by dislocation movement. So again, the relationship between dislocation movement and deuterium once again. Now, as I said, we get some surface cracking, and we were not really looking at the actual crack tip in that previous example. So Yasser wanted to try and find the real crack tip in the middle of the sample. So now the surface has been ground away to try and make sure we remove all of those surface cracks, and we're really looking at the main crack running through the sample. Unfortunately, we didn't manage to find the real crack tip, but this is an area that proved to be quite interesting here. So again, looking at our image in the SEM before we started doing the analysis, We can see this region here that crosses between these two cracks. We have grain boundary carbides along here, and we have a lot of localized plasticity in this region here. So again, no matter how we look at this image with different ratios, we can see we have very localized deuterium at the grain boundary carbides, and also again related to the localized plasticity here. So just to summarize all of that, those observations, in our in situ charging and deformed sample, we introduce some hydrogen into a CRAC sample. The stress that we're applying to it causes dislocation slip bands to form, and we can see deuterium localizing to those dislocation slip bands. The hydrogen is going to be transported by those dislocations, and they're really going to start to accumulate at those dislocation slip bands. And then what happens is our CRAC is going to propagate through those really hydrogen-enriched regions and propagate the CRAC further. Compare that to our pre-strain sample where we deformed it and then charged it without any additional straining. We do still see hydrogen accumulating at slip bands, but not to the same extent. So the other piece of work that I want to talk about was conducted by another student in the NanoSIMS group, Dr. Chris Jones. And Chris's work was mainly to look at zircaloy 2 and zircaloy 4 and investigate the effect of irradiation on the oxidation and hydrogen pickup of these alloys. But along the way, we discovered something that we were not expecting to see, and that was these hotspots of deuterium in the metal matrix. So just like I showed you before, we have an oxide layer, looking at it in cross section, and the metal underneath. And it was very clear we could see all of these hotspots. And if we have a closer look at one of these images, you can see there's a small hole in the middle of each one of them, just here. Now, these holes are actually the location of secondary phase particles in the zirconium, and we can very clearly see the hydrogen localizing around them. So we also worked with some collaborators who showed us or did some DFT modeling just to be sure that the deuterium was localized in the metal matrix around the SPPs and not in the SPPs itself. And that was confirmed by DFT. So this is what we're seeing. So we're finding that these SPPs are acting as preferential trap sites for the deuterium in the matrix surrounding the SPPs. And this is important because we're really interested in looking at deuterium pickup into zirconium alloys, and this had not been observed previously. So if we're really interested in knowing where the hydrogen is segregating to, where it's going in these zirconium alloys, we need to have a full picture before these alloys go into service. And so this was something that had not been observed previously. And I think this comes down to being able to look at a large area with the NanoSIMS compared to other techniques such as atom probe and TEM as well. So the real aim of Chris's thesis was to look at oxide growth during irradiation. So what we have here is two different images with different levels of irradiation on them. So on the left-hand side is where we started to move to try and image larger areas of a sample with the NanoSIMS. So this is actually just one strip of oxide presented over two lines here. And in this particular case, we had introduced oxygen 18 into the autoclave to look at where particular parts of the oxide were being particularly active in that time. So this alloy went through 131 days in an autoclave with normal water, and then it was transferred to another autoclave for 40 days containing oxygen 18. So in these ratio maps here, wherever we see pink regions, that's where we have a lot of oxygen 18 incorporation. So what this tells us is that the oxygen 18 has penetrated all through this outer oxide and can react strongly with the metal underneath. So we get incorporation of the oxygen 18 at the metal oxide interface. So we can describe this oxide here as being post-transition. So zirconium alloys go through a number of transitions where the oxide is either protective or non-protective, and whether the water can penetrate through that oxide or not. So interestingly, just how it happened with the area that we imaged, we found that this region on the side here and over here had not gone through one of these transitions. So we can show that the whole oxide does not undergo transition at exactly the same time point. Here, the oxide is still protected. Water cannot penetrate through it. We only get a small amount of oxygenating incorporation on the outer surface. So this is where we can show large-scale imaging. We're able to image over vast areas of the sample. And the key thing here with using isotopes as well is that without using isotopes, we wouldn't be able to determine which bit of oxide had just formed in that 40 days prior to it. If we look at samples in the TEM or atom probe, we may be able to see structural differences or slight compositional differences, but with the isotope, it's very, very clear over large areas of the sample exactly where the oxidation is happening at any particular time point. Now, over on the right-hand side, this is what we really set out to do, was to look at the effect of oxygenating incorporation after irradiation. So this alloy underwent a slightly shorter primary oxidation time. It was then sent to the Dalton Cumbrian facility, where Chris irradiated it with protons to a irradiation level of 0.75 displacements per atom. And then it went back into an autoclave containing oxygen-18 for another 40 days again. So in this case, we can see the oxygen-18 is mainly on the outer surface. So the underlying oxide over here is still relatively protective. But you can see that this zirconium alloy oxide is rather cracked because of this irradiation. And we can clearly see that water is penetrated down through those cracks, and it started to grow new oxide here using that oxygen-18 labeled water. We've got the cracks starting to penetrate down to the metal oxide interface. We can also see some very small regions here where the oxygen is able to penetrate through the oxide. It's starting to break down. So we get extensive cracking once we've irradiated the sample. And finally, just wanted to show you something completely different, just to show that the NanoSIMS can do lots of different things. This is some work carried out by Hannes, who was a DPhil student of mine when I was back at Oxford. She was looking at boron distribution in steels with different heat treatments. So on the left-hand side here, we have a as-quenched steel alloy, and on the right-hand side, an as-rolled alloy. And what you can see here in terms of boron distribution in this case is that there's very different boron distribution depending on the heat treatment. So in the as-quenched condition, we can see that the boron is primarily segregated to grain boundaries, whereas on the right-hand side, we can see boron localized to particles and precipitates. So this is another example of the NanoSIMS mapping over slightly larger areas. We can get much, much better statistics from the NanoSIMS than we can with TEM or atom probe when we're looking at, say, grain boundary segregation. We've got hundreds of grain boundaries or precipitates here. And this is all looking at boron at very, very low concentration levels. So much lower than you can actually achieve with SEM, for example, 0.02 weight percent boron in this case. So I'll just finish with some very sort of high level conclusions at this point. So we've been able to successfully image hydrogen trap sites in a range of different metals, steel, nickel, and zirconium alloys. And we've been able to do so at the scale of the microstructure as well. We can get some very unique information by using these isotopic labels. So in this case, I've shown you deuterium and oxygen-18, but we can also get very unique information on biological samples as well by using isotopes. So we can get temporal information about uptake and incorporation. So I hope I've shown that there's potential for NanoSIMS in many different research areas. And I think the key things to highlight from the NanoSIMS is that it's got high lateral resolution combined with high sensitivity. We can detect different isotopes, whether that's natural abundance or artificially spiked samples. And we can detect light elements. So we can do hydrogen, lithium, boron, which are going to be really important for a lot of material science applications going forward. So finally, I'd just like to finish with a few acknowledgements. In particular, I'd like to thank Yasser and Chris, whose work formed the real bulk of this presentation. Particularly like to thank Keshwe, who has almost single-handedly kept the NanoSIMS running over the last 20 months or so. And Keshwe is amazing at providing support for anyone who's interested in using the NanoSIMS. He's our experimental officer for us. I'd also like to thank David and Robert for working with them for the steel and nickel project, and Michael Preuss for the zirconium work. I showed you Hannis's work from Oxford. I'd like to acknowledge Chris Grosvenor there as well. Also EPSRC for funding Chris's and Yasser's PhD, and of course BPICAM for funding Yasser's PhD project, and Dr. Roberto Morano for supervising, being the BP mentor for the steel and nickel work. If you're interested in finding out the real versatility of what the NanoSIMS can do, you can use the QR code shown there. It will take you directly to the NanoSIMS website for our full publication list. And we also post all of our research papers on Twitter as well. And at this point, thank you for listening, and I'm happy to take any questions.